Chapter 5, Photosynthesis. Our world is based in solar dependence, as all of our food production is done by autotrophs, or organisms that can make their own food. The word literally means self-feeder, and we'll meet a lot of them over the course of the chapter. The algae, for instance, seen in B, is a massive oxygen producer, and it sits at the base of the oceanic food chain. The cyanobacteria in C produce a lot of oxygen as well. The plant in A can be called a photoautotroph. They use sunlight for energy and pull carbon dioxide from the air in order to create carbohydrates. We, like the deer seen here, are heterotrophs. We must eat other things to obtain our energy. Those other things are either plants themselves or animals who have eaten plants. Photosynthesis uses solar energy, carbon dioxide, and water to release oxygen and produce energy-storing sugar molecules. The reaction for photosynthesis, as seen here, 6 molecules of carbon dioxide, 6 molecules in water, and energy from sunlight react to produce 1 molecule of sugar and 6 molecules of oxygen. That sugar molecule, as written here with its chemical equation, is C6H12O6. That's the chemical equation for glucose. Hopefully it looks familiar. If you were to change this equation around, make it sugar plus oxygen equals water plus carbon dioxide, you end up with the exact same equation for cellular respiration. The two processes are mirror images of one another. When we look at photosynthesis, we always think about leaves, but not all cells in a leaf actually carry out photosynthesis. Cells within the middle layer are the cells that actually have the chloroplasts, which contain the photosynthetic apparatuses. You can kind of see a slow progression of close-ups. We're going to focus on the smallest one. Within an individual chloroplast, you have stacks of thalkaloids called granum, and the granums sit in the stroma. The light-dependent reactions, the first part of photosynthesis, takes place within those thylakoids. They have a specialized membrane. The products of the first part of photosynthesis head to the stroma. That's where the Calvin cycle takes place and sugar is actually made. If you go back just one little step, you can see the stomata, which shouldn't be confused with the stroma. The stomata is where the oxygen exchange occurs. Remember, plants have to take in CO2 and they end up making oxygen as a very helpful byproduct, and that's where it leaves. So about those two parts of photosynthesis, let's look at them written out so we make sure we've got it down. In the light-dependent reactions, we're focusing on the thylakoid membrane. Chlorophyll molecules absorb energy from the sunlight and they create chemical energy and oxygen. The oxygen leaves through the stomata and everything else heads to the stroma. This is where the Calvin cycle begins. Chemical energy is used to capture carbon and you form sugar molecules. Let's look at those two sections a little bit more up close. But first, remember, autotrophs can capture light energy from the sun, converting it into chemical energy, and we use that to build food molecules, right? Well, what is light energy? Light energy is the electromagnetic radiation that travels as particles on waves from the sun. Scientists have figured out how to quantify the amount of energy by looking at it in measured wavelengths. The longer the wavelength, that distance from crest to crest, the less energy is actually carried by that particle. Energy has been plotted on a color spectrum because we can actually see some of these colors, so it's just the easiest way to depict this phenomena. You can see the tiny, tiny section that's actually visible to us. Plants only absorb that visible light. We can use some of the other rays. Of course, you've all listened to the radio, and you've probably heard about X-rays as well. Now, there are different types of molecules called pigments that absorb these different wavelengths of light best. The wavelengths they cannot absorb physically bounce off of them, and that ends up being what you see. Most plants look green because their most common pigment, chlorophyll A, can absorb green light. The other common pigments are chlorophyll B, which absorbs red, orange, and blue lights, and carotenoids. So let's look at those light reactions. We'll start by studying a large complex called a photosystem, and we'll learn about two of them. The light reactions actually begin with photosystem 2, but that's simply because it was discovered second. So we start with sunlight being absorbed by the individual chlorophyll molecules in PS2, of which there are many. The energy captured is passed down a chain of chlorophyll molecules that ends at a reaction center. This reaction center gets so much energy that it actually releases an electron from itself. That released electron has to be immediately replaced, and it's replaced by one stolen from a split water molecule. Free oxygen is released and little hydrogen ions are created. That electron we stole is passed down an electron transport chain. The ETC powers pumps, protein pumps, that move the hydrogens to the inside of the thylakoid membrane. Eventually that electron is going to reach the end of the chain where it will meet photosystem 1. Photosystem 1 will capture energy. It doesn't split water again, but it does capture enough energy to pass that stolen electron onto a new type of cellular battery known as NADPH. When it's not charged, we call it NADP+, just like that NAD battery we met in cellular respiration. So we've built this big hydrogen gradient and we've charged some batteries. Well, we use that hydrogen gradient to power an ATP synthase, exactly like we did in cellular respiration. We create a whole bunch of ATP along with our charged batteries, and all the products head into the stroma. Let's do that one more time, just to be sure we're all on the same page. Light energy is absorbed by a chlorophyll molecule, and it's passed along a pathway to other chlorophyll molecules. The energy culminates in one chlorophyll molecule found in a reaction center. The energy excites one of its electrons enough to leave the molecule and be transferred to a nearby primary electron acceptor. That's just the first little electron acceptor in our electron transport chain. A water molecule is split so you can replace the electron you just lost. You lose an oxygen, or you don't lose an oxygen, but you create an oxygen and 2 hydrogen ions. You'll pair 2 oxygen together once you have 2 of them. That leaves the plant through the stomata. Those hydrogens end up being pumped into the inner thylakoid. Here's what that whole system looks like, spread out as kind of in mass. From photosystem 2, the excited electrons travels along the electron transport chain, and you end up pumping hydrogens into the inside of the thylakoid membrane. The hydrogens are allowed to leave out an ATP synthase so you can make some ATP. We also charge batteries, in this case known as NADPH. What do we do with all that stuff? Well, the light-dependent reactions are harnessing energy from the sun to make energy in batteries. Those products travel to the stroma, where the Calvin cycle can take place. What happens in the Calvin cycle? Well, we break it up into 3 stages. In stage 1, there's an enzyme, and we call it ribisco, and it incorporates carbon dioxide into an organic molecule. In stage 2, we reduce or remove electrons from that organic molecule using the energy that we created in the light reaction steps. We also use our batteries to help us to do that. In stage 3, a product known as RUBP is made, and we actually make quite a bit of it. We make 6 molecules of RUBP for every turn through the Calvin cycle. Unfortunately, RUBP, it's not really great. This Calvin cycle is not terribly efficient, to be perfectly honest. So for every 6 RUBPs that we make, by fixing carbon together and then kind of reorganizing all of its individual pieces, we only get to keep 1. So we make 6, we only get to keep 1. Where do the other 5 go? We actually have to use even more energy to turn the RUBP back into ribisco where we started. This is a cycle, so you have to keep having the enzyme products. And because you pull apart your enzyme, when you go through the carbon fixation process, it's not terribly efficient. I realized that this isn't anywhere near as in-depth as what you studied in cellular respiration. That's because this process of the Calvin cycle is so mind-bogglingly complex. We're going to keep it at this. This is as in-depth as we're going to go. In stage 1, the enzyme rubisco incorporates carbon dioxide into an organic molecule. So the enzyme rubisco takes CO2 molecules and it fixes them into this nice carbon molecule structure that we can work with. We attach 6 ATPs and that changes it into a different carbon intermediary. And then we reduce it. We take away electrons and we pass them on to something else. The energy for that process comes from all of our lovely batteries. After stage 2, we have 6 molecules of RUBP. Unfortunately, 5 of them have to be turned back into rubisco, but we do get to keep 1, and that's what the plant will use to make its sugars. Living in harsh conditions like out here in the desert has led desert plants to evolve different variations of this carbon cycle, and it essentially helps them to conserve water and energy. The issue with the carbon cycle, the issue with the Calvin cycle out in the desert is that you have to have a lot of oxygen exchange and carbon dioxide exchange, but every time you open up those stomata, you lose a little bit of water, and that's obviously, that's a big deal out here. So what a lot of desert plants will do is they will only open their stomata at night, or they actually have like little tiny, look like little tiny jail cells, or what you can think about when you walk into a store like Target. You walk into one door and there's air blasting, and then you have to walk into a second door. They have a special gated cell where one side of the cell will open, you let in some gases, shut the door behind them, and then you open up a secondary door, secondary opening. And this just allows control of the gases without losing a ton of water. So there are workarounds in this process. It does get really interesting, but we're going to keep it simple for our Bio 100. There are some different changes when it comes to the photosynthetic processes in prokaryotes as well, because you might be thinking, well, prokaryotes don't have organelles, so they don't have chloroplasts. How is this happening? Well, photosynthetic prokaryotes, they just have infolded regions of their plasma membranes that kind of mirror the functions of the thylakoids. So they can carry out this process without actually having a dedicated space for it. So it's not as efficient, but photosynthesis can still occur and they get enough sugars to survive. So the whole system is the carbon cycle, and you'll study all about it if you ever take an earth science class. But you have plants consuming carbon dioxide and releasing oxygen, and animals consuming oxygen and releasing carbon dioxide. And hopefully the big takeaway from this particular picture is that if you cut down all of the trees, you can't breathe. So please plant some trees. On that note, this chapter is over. Read your book, take notes in your own words, study very carefully. I again would encourage you to find other videos that discuss photosynthesis. It's very helpful to hear this from multiple perspectives. And then take a crack at your homework.